Biotechnology is the use of biological agents, living or parts of living organisms, for technological advancement. Aspects of biotechnology can be implemented to maintain a living stock of diversity, remove undesirable or disease-ridden traits, isolate metabolic products for therapy, increase yield, decrease costs, the ability for us to advance in biotechnology, the ability for us to apply these findings and facets to the wide variety of biological fields really depends upon two key things. One, the technology at hand, and two, our knowledge about the available living organisms and their utility. The first technique is DNA extraction. The goal here is to isolate and remove the genetic material from cells. To do so, you must break the cells to access the nucleus, break open the double-membraned nucleus, remove the proteins upon which the DNA is coiled, and then precipitate the DNA out of solution. The process of PCR involves manipulating a DNA sample using temperature to replace the roles of many enzymes. Heating slightly denatures the chromosomes, allowing the DNA to unwind. By cooling it, the DNA strand can be stabilized and the primers can bind. And then a slight temperature shift will stimulate the polymerase to bind and perpetuate elongation, because that last temperature shift is at its optimal temperature. The significance is that repeat cycles of this can be done on its own automatically and relatively quickly. You could get hundreds of copies in the span of a few hours. Once many copies of DNA are made, a likely logical step is to then sequence the DNA, determine the unique sequence of A, T, C's, and G's, the unique genetic code. To do so, you have to conduct a specialized PCR using some nucleotides that are radioactively tagged. And so a tube for each nucleotide base is used, one for the A's, one for the T's, one for the C's, one for the G's. Each of these tubes will also get primers, the same DNA sample, as well as a working DNA polymerase. And within each tube, we'll see that heating and cooling and slight heating shift cycle repeat. And so the DNA will unwind and the primers will stabilize and the polymerase will start copying. But when it adds a tagged nucleotide, the additional bulk from the radioactive label will result in the polymerase falling off. And when it rebinds and copies again, as long as it's adding regular nucleotides, it will continue down the sequence. But once it adds a radioactively tagged one, it will fall off again. And the result is that you get these different size fragments. These fragments coincide to location and place of that particular nucleotide in the genetic sequence. And we can determine this by running it on a gel, which will use diffusion to organize these sample fragments by size. Big things are heavy, and so they don't travel far, but small fragments are light and they travel very far in a short span of time. And so when you have this gel, you can read from the bottom up to determine what's the first nucleotide in the sequence, second, third, fourth, all the way to the end. This type of gel looks at nucleotide polymorphisms in the sequence. Another process can compare fragment length polymorphisms, and this is looking within specific gene areas where there are palindromic sequences that are subjected to insertions and deletions. And so using specialized enzymes to cut at these sequences will result in different size fragments within one or two particular genes, creating a unique banding pattern, which can allow us to look for similar patterns amongst closely related individuals or species. So in this particular example, we've got mom, her child, and 3 potential fathers. We can identify the bands that came from mom and then look to see which male can fill in those gaps. Similar process is done when we identify a criminal at the scene of a crime, or for immigration disputes, or identifying an unknown individual during times of war. based upon a small DNA sample. Another technique that is up and coming is genetic cloning. Not to be confused with organismal cloning, gene cloning is when we insert a gene of interest into a model organism. We could be adding a human gene for production of insulin into a bacteria because they're easier to grow and monitor. The process of this uses restriction enzymes, and that just means an enzyme that cuts the DNA at a particular place. And these cuts are at a diagonal, leaving sticky ends. So the DNA is much more likely to pick up surrounding genetic information to fill in the gap. And so you apply the gene of interest that you want, a little heat to stimulate the uptake and repair. Some of the bacteria or yeast will repair it perfectly without taking in the gene, but some will pick up the gene of interest. And those we would recognize as recombinant DNA. They've recombined an outside genetic source. In order to confirm who picked up the gene and who didn't, we typically add a tag to the gene of interest, either color or antibiotic resistance. With a color tag, those bacterial colonies will grow a different color. And so we know if you picked up the color gene, then you also picked up the insulin gene. Or if we looked at an antibiotic resistance gene, we would then grow a selective media culture. And so anything that grew had the resistance gene. And if they had the resistance gene, they also picked up our insulin gene as well. As we continue to expand on genetic engineering, it helps us to understand the goal of this process. It's not to meddle or play God. It's to help organisms become better suited to the environment or human use in a shortened time frame. Genetic engineering has always been a process through selective breeding, interspecies crosses, mutagenesis, transgenesis, and new breeding techniques like CRISPR and RNAi. Some of these processes happen in the wild with or without human intervention. And they all have their own level of human influence. But often when we talk about genetic engineering, people get nervous. There's controversy over genetic modified organisms. People are worried that they may contribute to an increase in food-related allergies, antibiotic resistance, cancer. They're worried that there's a monopoly on these genes and this technology, and whether or not we can control the spread of good genes or bad genes. There's the argument that if these things naturally happen in the wild, why should we be meddling in them? But the pros outweigh the cons. In agriculture, we can improve the nutritional quality of food. We can make it easier for farmers to grow with less fertilizer and water requirements. We can increase crop yields by relying less upon pesticides and herbicides. We can have large yields with minimal chemical intervention, which affects our water table. And in terms of safety, the FDA requires all genetically modified foods to meet the same requirements as anything else. And we've seen genetically modified technology help reduce the price of crops. The utilization of pesticides, which affect other wild plants and animals. Remember, genetic engineering isn't new. We previously called it selective breeding. We were engineering better plants. Native bananas are not edible because they have very, very large seeds. But by breeding and crossbreeding and breeding and selecting for bananas that had more fruit pulp versus seed, we have created the banana that you know today. Through selective breeding, the wild mustard plant has given rise to 6 different vegetables that we see in our grocery stores. Now, nature has done this on its own, but that takes thousands, sometimes millions of years. By selecting for the traits we want, humans can do it in 10 to 100. This allows humans to respond to an environment that's changing at a much more rapid pace. All of this is possible because the genetic code is nearly universal. It is shared by the simplest bacteria to the most complex animals. And so A, T, Cs, and Gs, that order of a particular gene can be moved and transcribed and translated in any species, so long as the instruction is there. Here we see bioluminescence from a cyanobacteria transplanted to a plant and a pig, showing just how universal this instruction is. With controlled breeding programs, nature doing most of the work, humans with a little direction and influence, we have been able to cross and create many diverse dog breeds by identifying the desirable trait we wanted and crossing the pups that had that trait until we got what we want every single time. The downside of controlled breeding, though, is that this takes a lot of time, and it's going to include a lot of intermediates, near hits, near misses, and you don't know what you get because genetic recombination changes the entire genome of those animals. Genetic engineering, however, moves 1 to 4 genes only. We know exactly what we're moving and where we're putting it. It's instantaneous. It's in the next generation or it's not. No intermediates, no hits and misses. We can check for it. And so genetic traits have been approved in close to 400 plants worldwide and about half of them in the United States. We've made canola that are herbicide tolerant. We've made papaya with disease resistance. We've made soybeans healthier by adding high oleic content and increased omega fatty 3s. Where we can move one gene from a plant to another plant has opened the doors to gene therapy. Could we move functional genes from one human to treat or prevent disease in someone else lacking that genetic instruction? Could we replace a mutated gene with a healthy copy? Could we introduce a new gene to help fight disease? There are 2 types of gene therapy. Know the difference between ex vivo and in vivo. Vivo means life. So ex vivo means outside of the body, outside of the living organism. The cells are removed, they're manipulated, and then they're re-put in. In vivo means all of this is happening within the body. The gene therapy is introduced to the person and then has to navigate the entire organism system to find the specific cells of interest, target those genes, and replace them. How can we achieve this? By using stem cells to repair damaged tissues, or using viruses, because they are, at their core, genetic delivery mechanisms. Gene therapy is not the end-all be-all cure at this point in time. It's limited to somatic cells and disorders that are caused by a single gene. But that still opens the door to a number of diseases having treatment and potential curative options where before there were none. And ultimately, this is an alternative to a lifelong prescription of drugs or a very invasive surgery. As futuristic as genetic engineering is, biotechnology can also be quite simplistic. Antibiotics is an application of biotechnology where we use chemical agents to treat microbial infections. Antibiotics can either kill the microbe directly or prevent them from reproducing. Vaccine technology is another application of biotechnology, wherein we're able to introduce a small amount of the virus or bacteria to teach the immune system, to prepare a memory response. So that way, when the real threat is encountered, our immune system is prepared and able to mount a very effective counterattack from day one. Vaccines are considered the gold standard because they're the only therapy that potentially can prevent disease from occurring at all. Given that vaccines are relatively old, a lot of people have asked, well, are they even still necessary? Yes, microbes exist in all parts of the world. Some will say, well, when I was a baby, there were only a few shots. Now my kids seem to get so many. That's because with biotechnology advances, we have more vaccines available to us now, which gives our children more protection than we were afforded. There have been some concerns about vaccines being safe and potentially causing autism, but that has been disproved by over 250 independent studies. Autism is caused by a genetic component. What about side effects? They give you a long list. Those are minor in comparison to possible death, which can come from a number of these diseases. And some wonder if this is a ploy by Big Pharma to make money off of you. Keep in mind that most vaccines are covered by your insurance 100%. And so if you really want to stick it to Big Pharma and the capitalistic man, get your vaccine, because that's how you save money. If you're hospitalized, even the best insurance policies only cover about 80%, and even a single night stay can run in the thousands. One of our newest technological applications is RNAi. The I stands for interference. The goal here is to use small interfering RNA, small segments of RNA, to bind to similar sequences of your own native RNA. And when they become double-stranded, this activates our body's recycling protocol because RNA is never meant to be double-stranded. So the enzyme dicer recognizes a double-stranded RNA as dysfunctional and degrades it, breaks it down to little pieces. This prevents that RNA from ever being translated and expressed. The idea is that RNAi could serve as an anti-HIV cure. What are the ethics behind the application of biotechnology? Well, the guiding principles of this effort is to do no harm, but more importantly, to do good for people, not violate the individual freedoms, and to be fair across all people and walks of life. Secondary to this is about transparency, keeping promises, and protecting the information.